All right, we're ready to start 7.2. So let me get on 7.2. The essential idea is this idea that we're taking DNA and we're copying it into RNA. And remember the general idea, DNA gets transferred into mRNA, gets transferred into a protein. And these are all basically types of information. DNA is like the general storage area, and protein is like the practical application of that knowledge. Now, kind of the big picture here is this really interesting idea of epigenetics. Normally, when I was taught biology, whatever happens to you individually during your lifetime does not get passed on through your gametes via your sperm or your eggs. But we know now that that's not necessarily true. Normally, when the sperm and egg fertilize, everything that happened to that DNA during the lifetime kind of gets wiped out and reset. But scientists have determined about 1% of the impacts that have happened on that DNA over time do affect the embryo, do get through the patterns of changes that occur. And what I'm talking about there is mostly this idea of methylation that we'll get into. Right here is a cytosine. Remember, you have A, T, C, and let me go ahead and do the proper number of bonds. Cytosine is one of those nitrogen bases, and what happens is a hydrogen right here is replaced with a methyl group. Remember, just meth is one carbon with 4 hydrogens, and what that does, it kind of turns off that DNA, and it makes it so that DNA cannot be transcribed. So in the DNA itself, what you see this diagram on top, this is unmethylated DNA here. It has acetyl groups on the histone proteins, which are these 8 proteins that the DNA is wrapped around. And everything is good. It's turned on. The gene is turned on. Therefore, it can be transcribed and turned into a protein. Down below, what we see is these methyl groups. Methylated C's, basically. And it's deacetylated. And it's basically turned off. Histones, correct that. Gene turned off. So these turn-off events, these methylated areas, normally they do get reset and reprogrammed, so to speak, but some of that does get through. One example would be gestational diabetes. If a female gets gestational diabetes when she's pregnant, those high levels of glucose that are circulating in the mother's blood can get into the fetal blood and therefore methylate some of the genes in the fetus and make that fetus, when it turns into a human being, have gestational diabetes in its lifetime. So something that's affecting the mother during her lifetime can be passed on to the offspring. So back up a little bit, just about transcription. Very handily, it occurs in that 5' to 3' direction, just like DNA replication that we talked about in the last video. Oops, So this is the direction of synthesis. I almost wrote replication because I just said that word. So direction of synthesis or direction of translation, or I'm sorry, transcription. Remember transcription, because those get mixed up all the time, like I just did. That's DNA to RNA, specifically messenger RNA. Translation is taking that messenger RNA and making the protein. This top strand would be called the non-template. You see it's not actively being used. To match up those bases to make the DNA, or sorry, make the RNA, the messenger RNA. Let's just clarify this. This pink right here is mRNA. Yeah. MR. And I gotta slow down. There we go. And of course, this non-template, you can imagine this part of the strand is called the template strand. It's actively being used to match up the RNAs. And if you do notice, the non-template strand, it matches. So these part right here, they match. Except for, right, you have U's on the messenger RNA and you have T's, but otherwise they match up. They are the same. So that might help you at some point when you're working on your biology. So again, this comes back to that nature of science that we talked about. If we imagine this little area right here is the gene that we're trying to use, to make some certain protein. These are the methyl groups, the methylation that we see, and these are the histone tails that are poking out. We mentioned those histone tails. We looked at those tails and that using that visualization software. So we can't get the DNA. The gene is inactive, so this gene is not expressed. It's not seen. It's basically hidden. The body can't use it. Down below, we see, oh, look at how wide open that gene is. So these, with these acetyl groups, this gene can be expressed. If you just look at the visual, if you just look at the way the structure is, doesn't it look like that bottom one, the gene is open? RNA polymerase can grab onto there and make that messenger RNA. It's pretty obvious. You look at the top, it's buried in there and the gene is inaccessible. So two types of chromatin. Remember the chromatin, that's the DNA plus the protein. Let me switch up my color. DNA plus protein. And remember that protein, it's called a histone protein. And another, we see these areas with a lot of acetylation. And that's areas that can be transcribed, basically turned on. And these areas with methylation, it's turned off. So that's what scientists are looking for when they're looking for areas with high gene activity. They're looking for these areas with acetyl groups versus the methyl groups. Determining, what is actually, being made on this gene. So after the RNA, the messenger RNA gets produced, it undergoes a lot of processing in eukaryotes. And I talked about in the last video, we have these exons and introns. Remember, the introns are not used, so they get cut out. The exons go into that final product. So this is our original RNA. And then it goes with this middle structure. This middle diagram is showing the finished messenger RNA, or the nearly finished messenger RNA. You have, this is with the exons removed, or the introns are removed in that first step. So now you just have the genes or the code that's going to be expressed. But you see, oh, what's this stuff? Okay, this is called, capping. One side of the gene is capped off, and then in the middle we see it's being spliced. That's what this is called. And then we have polyadenylation, or poly-A tail. Basically just a whole bunch of A's, just a whole bunch of adenines. And this is our processing. So a lot of work goes in after that messenger RNA is produced to get it ready to then go out into the cytoplasm, grab on to a ribosome, and then start making the actual proteins. So here we go, DNA, RNA. We see there's a bunch of different ways. You might think that splicing, that's kind of a hassle, but by doing that, there's a whole bunch of ways we could arrange our messenger RNA to make a whole bunch of different proteins. So from that same information, if you change the splicing techniques, you can make a whole bunch of different products. One of the reasons why humans don't have the most genes. Scientists originally thought we had like 100,000 genes, maybe more. It was originally, now I believe it's about 24,000 genes. Check that. Maybe 22,000 genes. That's less than other things like an earthworm, I believe, or fruit fly. But we can be more sophisticated than those living things because we have this ability to really highly control and modify the splicing. You get a whole bunch of different proteins. So this, say protein A, I'll go ahead and write it out. Protein B, protein C. All from those different ways of splicing up the product that we have.